Good day, this is Professor Will, CBMD PhD. Today's Thursday, March 10, 2022. It's 3:20 and 52 seconds, a.m. Eastern Standard Time. Today's research, Polycystic ovary syndrome. So let's hop to it here. Anti-Mullerian hormone may contribute to infertility in polycystic ovary syndrome. Date March 9, 2022. Source, Weill Cornell Medicine. High levels of anti-Mullerian hormone have traditionally been thought of as merely a passive byproduct of polycystic ovary syndrome, but a new preclinical study suggests that the hormone plays an active role in the disorder and may contribute to problems with ovulation and fertility. The investigators discovered that anti-Mullerian hormone may cause follicles of multicellular fluid-filled sacs that contain developing eggs in the ovary to mature too quickly. Anti-Mullerian hormone is routinely measured in the clinic to give an indication of how many follicles a woman has growing in her ovaries, and this value is often high in women with polycystic ovary syndrome. But no one has ever determined whether a high level of anti-Mullerian hormone by itself can have a negative influence. A better understanding of the root causes of polycystic ovary syndrome is critical to mitigating the condition's many health consequences. Developing eggs, called oocytes, are produced in the ovaries within follicles, which support their growth. As the follicle grows, the egg that resides within it gradually matures, and, in a normal reproductive cycle, that follicle ultimately undergoes ovulation to release an egg capable of being fertilized. However, in patients with polycystic ovary syndrome, a hormonal disorder that impacts up to 10% of women, the ovaries contain numerous smaller follicles that produce anti-Mullerian hormone, but fail to progress to later stages and ovulate. While fertility treatments can help with conception, women with polycystic ovary syndrome contend with a variety of other symptoms. For example, polycystic ovary syndrome is often accompanied by hyperandrogenism, or high levels of hormones, such as testosterone, that are collectively called androgens. An overabundance of these hormones can cause an array of secondary complications, including unwanted growth of body and facial hair. Women with polycystic ovary syndrome can also be prone to diabetes and metabolic disease and are more susceptible to endometrial cancer. The research team hypothesized that anti-Mullerian hormone is directly contributing to at least some of the constellation of symptoms associated with the disorder. And to isolate the effects of anti-Mullerian hormone, the researchers utilized a xenograft system in which ovarian tissue from human organ donors is engrafted onto the flank of immunocompromised mice. One group of mice was transplanted along with cells that continuously supplied anti-Mullerian hormone directly to the grafted tissue, and the other group of mice was transplanted with control cells without anti-Mullerian hormone. What we have here is a unique opportunity to take tissue from the same human donors and allocate it to both experimental groups, the group with anti-Mullerian hormone and the group without anti-Mullerian hormone. Researchers say this is actually the best control you can hope for in these types of experiments. By contrast, when researchers compare patients with polycystic ovary syndrome to those without the disorder in clinical studies, a variety of influencing factors, such as genetic predispositions and varying levels of different reproductive hormones, must be considered. The researchers discovered that the ovarian tissue exposed to high anti-Mullerian hormone contained follicles that showed characteristics evident at a much later stage of development, and specifically, the follicles were undergoing luteinization, a process that occurs immediately leading up to ovulation before any of the oocytes were ready. Anti-Mullerian hormone is causing the usually coordinated growth process between a follicle and its resident oocyte to fall out of sync. It is like baking with the oven too hot. The outside, or the cellular component, of the follicle is overcooked, while the inside, or oocyte, is not done. based on ultrasounds showing persistent presence of multiple small follicles in the ovary. Infertility in women with polycystic ovary syndrome has been attributed to stalled growth and resultant failure and egg maturation. But recent data indicates that stalled follicles in these patients are actually a steady stream of newly growing but abortive follicles. And this new study puts forth the revised interpretation that the persistent array of small follicles in polycystic ovary syndrome, ovaries, represent a continuous succession of follicles undergoing rush maturation and failure to ovulate. Considering the broad prevalence of polycystic ovary syndrome, the factors that contribute to its emergence remain poorly understood. Although treatments exist for individual facets of disease, the condition rarely recedes until women reach the end of their reproductive lifespan. The identification of a novel anti-malarial hormone-driven mechanism for pale follicle development suggests that it can contribute to this and perhaps other facets of polycystic ovary syndrome disease symptoms. With further research, researchers hope to develop drug treatments that target the effects of anti-malarial hormone, which can help restore the menstrual cycle in women with polycystic ovary syndrome and manage other symptoms of the condition. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoy listening. Until next time, have a great day.